organism, we came across many such things, and one among them was in unicellular organisms. The cell division and the reproduction is mutually inclusive. That means what? The reproduction which takes place in the organism, in the unicellular organism, the reproduction which takes place in the unicellular organism is nothing but the cell division. Okay. The cell division itself is the, hold on, guys. Okay, so the cell division itself is the reproduction. See, you can see over here, right? So what happens over here? That one cell, it divides. Okay, that's one cell. It divides just now when you have seen it with the one cell, it divides to give out the other cell. Okay, it divides to give out the other cell. And in unicellular organism, the cell division itself is a reproduction. Okay, they reproduce by the asexual reproduction method. Okay, they reproduce by the asexual reproduction method, and the cell division itself is a reproduction process. Okay, whereas a multicellular organism, cell division is different and reproduction is different. Okay, they reproduce differently. They reproduce differently, and the cell division is different in the multicellular organism. Okay, in the multicellular organism. Now coming to the cell division in unicellular organism can be observed in vitro. Okay, can be observed in vitro in test tube, petri dish, or under microscope. Okay, as you all are aware, when we were studying about the introduction to biology, we had come across that we see the unicellular organism or the microorganism only under the microscope. Yes or no? Everyone knows that, right? Who was the first person to see the microorganism in the microscope? The first scientist to see microorganism in the microorganism in the microscope. Anyone remembers? Anyone remembers it? Ah, yes, very good, Sharad. It is Anthony van Robert. Sorry, it is Leeuwenhoek. Anthony van Leeuwenhoek. Okay, Anthony van Leeuwenhoek was the first, not Robert Hooke. It is Anthony van Leeuwenhoek. Okay, so Anthony van Leeuwenhoek was the first person to see the microorganism. But very good. Remember that. Remember at least the who. So it is nothing but Anthony van Leeuwenhoek was the first person to see the microorganism under the microscope. He had seen that in the rainwater. Okay, very good. So, Anthony van Leeuwenhoek was the first person to see the microorganism under the microscope, and we see these microorganism only under the microscope, whereas we cannot see it in the naked eyes. Okay, now cell division in unicellular organism can be observed in vitro. In vitro means inside. Okay, inside means in labs. Okay, in vitro in a sense in labs, in laboratories we can see that. So as we see this in vitro, that is in labs, we can see that we can see it in the test tube. We can see it growing under the test tube, petri dishes, and or under the microscope. Okay, we'll know more about this, how we can see that. Before that we will see what is cell division. Can you see? Everyone can see here there is a cell and it's getting divided to get to have a different cell. Okay, the one cell gets divided, one cleavage is formed here and it forms another cell. Everyone can see here for your better understanding. Please, so that you can understand how the cell division takes place. Okay, so what happens over here? You have an idea, right? The other day when I was saying you that one cell gives out two cells. So how does it happen? This happens in this way. This one cell, okay, this one cell forms a furrow in between. Okay, forms a furrow or a gap in between and gives out the another cell. Okay, gives out the another cell. The same way it was here as well. You must have seen this here in the last slide. Can you see it over here? This one cell gives out the other cell. Okay, this one cell gives out the other cell, that is nothing but the cell division. Understanding? Understood this? Okay, this is in the animals. What happens is this one cell having two nucleus here. Okay, what happens? This one cell forms two nucleus and they give out a two cell. One cleavage furrow is formed in between. Whereas in the plants also this takes place, whereas one wall-like structure also is formed. It is we'll know more about plants, how does it happens and all in the meristematic tissue. You must have come across as well when you are learning means doing the assignment. So now in here, what happens in the animals? Let us understand this first. So what happens is this one cell gives out the two nucleus and the cleavage furrow is formed, giving out the two different cells. Okay, giving out the two different cells. Understood about the cell division? Anyone has got any doubt regarding this? Clear about the cell division, right? So like can you hear me now? Sorry for the inconvenience, students. Sorry for the inconvenience. Uh, Sorry for the inconvenience. So this is about the cell division. Can you hear me now? Am I audible? Okay. So let us move on to the topic which we were discussing, that is about the cell division. Okay. So hopefully we have understood this. Sorry for the scribble. There, there is an unstable network, students. I'm so sorry regarding this. Okay. Understood, right? How the does the cell division takes place? Got an idea, so that it becomes easy for you to understand. Now is it proper? Okay. So now, as we have finished about this cell division, let us move on to the. Okay. So let us move on to the next thing, that is about the in vitro. Now is it audible? Is it audible now? Okay. Okay, fine. So as it's audible, if there is any such issue, please do tell me. Okay, thank you. Yeah. Now coming to the in vitro growth, which we were talking about, the in vitro cell division, which takes place. You must have come. See, I will tell you where we have come across this. That is about the unicellular organism. Okay, the unicellular organism where here. Okay. So unicellular organism can be observed in vitro, that is in the laboratories, that is the test tube, petri dishes, and under the microscope. Now you have got to know this. So now let us know that where actually and how does it occur. Okay. So in the as in the petri dishes, so this is in the petri dishes, how these microorganism looks like this. Okay, the microorganism in the petri dish looks like this. Okay, that what happens in the laboratory in the agar broths or any nutrient media, we grow this microorganism. Okay, we grow this microorganism. Can you see over here? Like this, they occur in this way. Okay, the growth of the microorganism in the petri dishes. These dishes are nothing but called as petri dish. Okay, there will be a covering over it. We have to seal this with the paraffin wax, and what happens is we let the microorganism grow. So in vitro that is inside the lab, what happens is in the petri dishes in this way the microorganism grow. Okay, the microorganism grow. Is it clear now about the microorganism? Yes, how they grow in the petri dishes. So this is for you to understand. So how the growth in the petri dishes of the microorganism takes place. Okay. Now coming to the next one. Coming to the next one, that is how you see here is the same way the petri dishes the bacteria is growing. Okay, in the petri dishes the bacteria is growing. So in the laboratories we grow this bacteria, microorganism, any such fungi. Okay, any such fungi, these are grown in these petri dishes. In the nutrient broth, what happens is the growth of the microorganism takes place. Here is the bacteria. Whereas also in the test tubes also we grow microorganisms. Okay, what we do is we make a slant of this, a slant in this way. Okay, in this way a slant is made of a nutrient broth, and what we do is we allow this microorganism to we allow this microorganism to grow here. Okay, what happens here? As the microorganism grow, there is a chances of the color changes taking place. Okay, color changes taking place. The bacteria, the fungi, or any such microorganism which is growing in this test tube can give out this color to this nutrient broth. Okay, so give out this color to the nutrient broth, and these are the pet, different test tubes which are used for the growth of the microorganisms. Okay, for the growth of the microorganisms. So in the laboratories, how in the petri dishes and in the test tubes, the microorganisms are grown. You got the idea, right? How these microorganisms are grown in the laboratories? Anyone has got any doubt regarding this? How in the laboratories, how they are grown, that is in vitro. In vitro is nothing but inside. Okay, in vitro is nothing but inside. So inside how they are grown using test tubes, they are grown using petri plates. Okay, so these are the petri plates used and these are the test tubes. Okay, clear, right, about regarding the topic? Okay, so Reena, what it is microorganisms. Microorganisms, which if we want to grow it inside the laboratories, what we do is we use this petri plates. Okay, and these test tubes. So what we do, we use the nutrient broth containing any such carbohydrates, sucrose, sugar. Okay, sorry for the inconvenience. Okay, sucrose, sugar, or potato dextrose, or anything is used. Okay, any such nutrient media is used. Okay, so what we do is we allow this nutrient media to we pour this nutrient media in this petri plates. Later, what we do, we allow the microorganism, we put the microorganism inside this petri plates having the nutrient broth, and what happens is the growth of the microorganism takes place. We seal this petri plates with the paraffin wax. Okay, paraffin wax is we seal it so that other microorganism or any contamination should not happen. So what happens here? So in vitro, in this way the microorganisms are grown inside the laboratories. Okay, inside the laboratories, how they are grown? We use a petri plate, using a nutrient broth, we allow the microorganism to grow here. Okay, we allow the microorganism to grow here, and inside the laboratories, in this method, using petri plates or using the test tubes, we can grow microorganisms. Okay, so here in the test tubes, what we do here, see there is a stopper here, the stopper or a cap. We use it for the test tubes. Then later we allow the nutrient broth to we pour the nutrient broth inside the test tube in a slant manner. Later what we do, we put the microorganism inside here and using the nutrient from the test tube from the nutrient media, they grow. Okay, in this way we can grow the microorganism. Okay, in this way we can grow the microorganism. And how the microorganism grow? Microorganism grow by cell division. Okay, the division of the cell is only the growth of the microorganism. Okay, is it clear? The topic is clear? Okay. Next coming to everyone. So everyone it is clear? Okay, very good. So next coming to the so here now as we are discussing about the living organism, we have discussed about the different how the cell division takes place, how the reproduction takes place. Okay. So next coming to the difference of growth in non-living objects and living organisms. Okay. How does the difference of growth in the living objects and living organisms? So as you all know, living organism, how does the growth takes place? How does it takes place? The growth in the living organism. How do they grow? How do they grow? How do they grow? Tell me. Cell division. Very good, Purnima. Cell division. Yes. They grow by cell division. Yes, good Sonal. They grow by cell division. And in now we will know about how in the non-living objects the growth takes place. Okay, growth in a sense, let us know what it is. Yes, very good, Karthik. Yes. Cell division, yes. Divya, Monica, yes, very good. So in both, increase in the mass occurs. So in the non-living, that is abiotic, and in the living, that is biotic, in both, increase in the mass occurs. Okay, increase in the mass means there is some growth which is taking place, by which we can say that, okay, this living organism is growing. Okay, this living organism is growing. I have given you the examples in the last class as well. So what happens is, from a very small toddler to an adult, what happens, there is a growth which takes place, that is increase in the mass, increase in the size of the organism, or that particular human being. Yes or no? That is nothing but called as growth. You measure growth by saying that growth increase in the mass and the size. Okay. Now what happens in non-living things, in non-living objects? How do you just say that, okay, this growth is taking place, or increase in the mass is taking place? Okay. So how does it occur? Is increase in the size, increase in the size of crystals, snow mountain, deserts, etc. are the example of growth. So what happens here is increase in the size of the crystal, snow mountains. That is nothing but the increase in the rock size, or the increase in the sand of the deserts, or any such thing. We say that non-living, the increase, non-living things, objects are increasing. Okay, these are the example of growth for the non-living things. This results for the accumulation of the materials on the surface. That is occurs externally. So what happens here is, any mountain. Okay, for example, take a mountain or a rock, take a mountain or a rock. So what happens is the accumulation of the sand over it, or the accumulation of the stones, the rocks over it. Okay, what increases the mass of that particular mountain. Okay, the accumulation of the sand, rocks, stones over it increases the mass of the mountain, and that is nothing but is called as a growth in non-living objects. Okay, that is considered as a growth in non-living objects. There is no such cell division or anything takes place. Okay, there is no cell division takes place, whereas just the accumulation of materials on the surface. Okay, accumulation of the materials on the surface is nothing but called as growth in these non-living organism. Whereas the living organism, growth occurs internally. Okay, growth occurs internally. Many metabolic activities takes place, cell division takes place, and because of what we grow. Okay, because of which we grow. The living organism growth occurs internally. Okay, cell division takes place internally of a body, which results in the increase in the mass and the size of the body. It is not just the accumulation of the matter of the body. Here we grow by cell division. Okay, there is no accumulation. In living organism, there is no accumulation. We grow by cell division. The division of the cell takes place, and because of what we grow. Okay, whereas in the mountains and all, the accumulation of the sand, stones, rocks, or anything as such results in the growth in the non-living objects. Growth is irreversible and is characteristics of the living organism. Once you grow, you cannot come back to a small. Once you become an adult, can you come back to a small toddler, or come back to as a small baby? Is it possible? Anyone? No, it's not possible, right? Once you are fully grown, it is not possible for you to come back to a very small baby. So what happens here is the growth is irreversible. Once you grow, you cannot come back to a smaller version of yourself. Okay, and growth is a characteristics of a living organism. Okay, the living organism only grow by the cell division and all, whereas the non-living objects, they just accumulate and they increase in the mass. Okay, they just accumulate these materials, non-living materials, and they increase in the mass. Whereas the human beings, what they do, the cell division takes place, because of which the mass, they increase in the mass and increase in the size. Okay, Clear to everyone? Anyone has got a doubt regarding the difference of growth in non-living and objects and living organisms? See, there is no confusion to make. Just remember this. In non-living objects, in non-living things, what happens is only the accumulation. Okay, only the accumulation of the this object, accumulation of the matters takes place. That, for example, I gave you mountains. What happens is on the mountains, sand accumulation takes place, stone accumulation takes place, and many things accumulate and they become a bigger mountains. Okay, they become a bigger mountains. Whereas they only increase in a mass. Okay, whereas they only increase in a mass. They do not divide. Okay, they do not divide. But whereas in the human beings or the living organism, what happens is we divide, cell division takes place because of which we increase in the mass and the size. Okay, we increase in the mass and the size. Do you have any doubt regarding this particular thing? Any topic which we have done today? Just we'll go once again back because there was a lot of disturbance. So this is about the microorganism growing in vitro. Okay, that is in the Petri plates and in the test tubes. Okay, how you have to know, right, that how does the this microorganism grow in the laboratories? This is the way. Okay, so next is, now if I ask you in the next class that how does the this microorganism and microorganism grow in the laboratories, then the answer will be, how does the microorganism grows in the laboratories? Then the answer will be, how does it grow? Where do it grow? In what? No, cell. How we grow? Yes, very good, Sonali. In the test tube and yes, vitro culture. In vitro culture, it is. Where the, how does it take place? In test tube and there is one more thing I said. Petri plates here or petri dishes. Okay, petri plates or petri dishes. Yes, very good. In Petri plates and test tube, we see this microorganism growing. Okay, we see this microorganism growing. And we can also count the microorganism in the microscope. Okay, using microscope, we can also count it. Yes, very good. Test tubes and Petri plates. This is how the cell division takes place. That is from one cell, another cell comes out. Okay, and this is about the, is there any doubt regarding this topic which is done today? Any doubts?